And the recording has begun. So it is, for everybody, September 26th. This is the first of 2 lectures on more of a psychopharmacology focus, broad overview, and then we'll narrow it down as we go along in the remaining units. Any questions about any of this material, stop me, okay? It's pretty dense and pretty technical, so pause if you need. Take lots and lots of notes. All right, Karen's ready. She's ready to roll. So a lot of this is basic. This is, well, I say basic, maybe I should say fundamental more so. Starting at the start, right? So the central nervous system, basic division of neuroanatomy. You're going to have the central nervous system and you have the peripheral nervous system. And the central nervous system is pretty easy. It's your brain and your spinal cord. Tidbits about your brain that SATIC talks about, cocktail party kind of information, that your brain weighs about 3 or 4 pounds. Not a whole heck of a lot relative to the rest of you. 100 billion neurons.
I don't feel like he sees you.
But actually, now.
Oh, hey. Hey, everybody. If you're logging in, we're thrilled that you're here. Just please mute your mics because we can hear everything. Every single thing. So this is an exercise that I like to do if anybody has an iPhone with a calculator. This is fascinating to me. If you can calculate 1 million seconds, how many days, weeks, years is that? 1 million seconds. It's worth the effort. It's worth the effort of doing it. Gold star to anybody who can calculate that in lightning speed. Let's see who the mathlete is among us. And maybe that's actually just too nerdy of a task, but yay! Mathlete award goes to Deborah Jones. Thank you, ma'am. Yes, 1 million seconds. 1 million seconds is, ooh, wow, that's serious right there. Yes, ma'am. 1 million seconds is about 11.5 days. So let's say you were born with 1 million neurons. And let's say in utero, you made your brain, your DNA was able to manifest 1 neuron per second. Then in utero, it would take, okay, well, 1 million seconds translates to 11 days. If I made 1 neuron a second, it would take 11 days to come up with, generate all those neurons. In utero, you got 9 months. In contrast, 1 billion seconds, a billion seconds, equates to 30 years. And you're born with 100 billion neurons. So if your brain, if your little DNA protein factory was pumping out a neuron a second, it would take 30 years to arrive at 1 billion, much less 100 billion. So that should speak to the, cosmic speed with which neurons, with which your brain is developing in those 9 months. You got 9 months to make 100 billion neurons. If you're making 1 a second, it would take you 3, what would that be, 3,000 years? Make it, so think about if a child in utero is exposed to substances, is exposed to some nasty virus, is exposed to systemic inflammation. No wonder it is a really detrimental, deleterious event for developing brains. Because you got to end up with 100 billion. And boy, there's a lot going on. So if you miss a window, if you just, so the value going forward of talking to people about avoiding exposure to substances, especially to moms, to pregnant women, even cigarettes, even reducing blood supply. Briefly, it just blows my mind. Yeah, 1 billion seconds is 30 years. Speaks to how much more that is. So basic terminology, things you need to know that probably aren't going to be deal breakers. Different names for different kinds of neurons. You got ependymal cells. You remember those? They produce cerebrospinal fluid, so those are important. You got astrocytes. They kind of look like stars. They attach things and keep things steady and connected to each other. Oligodendrocytes are really important because their job, down here you see myelination. Myelin is a very fatty, a insulation for neurons. And it's a cell that it wraps around and around and around the axon, which is the pipeline from the head of the neuron to the business end of the neuron. Energy has to travel down that. Electricity has to travel down that. You want it to be as efficient as possible, so it gets wrapped with a ton of just the right amount of myelin, which reduces signal loss and makes for speedy, efficient transmission of neuronal impulses. There are little gaps in it called nodes of Ranvier, and there are diseases of that on inspection. You can see loss of myelin. Demyelinating diseases affect the efficiency and even the ability of nerve cells to communicate with each other. Interestingly, the frontal lobe of the human brain is the last structure in the brain to get its final wrappings of myelin. Is that any surprise? No, it's not, because when you look at the function of the frontal lobe, which is things like sustained attention and predicting consequences and impulse inhibition and planning and complex sequencing of events, those are skills that people hopefully develop by the time they're in their early adulthood. Any of those executive functions are not pronounced in adolescence. Well, the connectivity of that structure, that executive structure, to the rest of the system is incomplete until that point. All the more reason to not expose young brains who are trying to lay down the infrastructure for effective high-order existence and cognition and functioning. You don't want to mess that process up. No surprise. So that's the brain, our brain. And then there's our spine, which is how the brain's, how it's going to communicate all the way down to your sacrum, and then any innervation that leaves the central nervous system, so any outgoing motor neurons, it all goes up to central command up in the brain. You have outgoing efferent neuronal impulses. So efferent means exiting. Efferent exits the brain. Afferent approaches the brain. So sensory information from thermoreceptors on your skin, that all has to travel in an afferent sensory neuron to get up into the thalamus to be recognized and processed. Anything outgoing has to travel an efferent pathway. And if you remember from those, and I think Sadek has it, or I know you had this in physical assessment or pathophysiology in your first year, but you have your incoming and outgoing locations of the spinal cord. You have the dorsal horn, you have the ventral horn. If you remember all of that kind of information, that there's distinct pathways exiting to cause things to happen, incoming to make its way up to the brain. In just terminology, you'll see a tract is the same thing as a neuron that's in the central nervous system. It's not called a nerve, it's called a tract, and where a bunch of them group together, maybe to make a node or a little hub. It's just called a nuclei. So when you see, the thalamic nuclei, they just mean it's multiple neurons meeting together, communicating with each other, a point of intersection. So it's probably an important little location as well. So that's all that means. You don't call nerves in the central nervous system. It's a tract. Whereas in the peripheral nervous system, this is really everything other than your brain and your spinal cord. Really important. I mean, this is where outgoing information has to go to command every bodily function, from bladder control to moving your pinky finger to, all of it. All peripheral nervous system. And it's divided into different branches, right? You have the somatic branch. Then you have the autonomic branch. So which of those is involved in voluntary activity, somatic or autonomic? Autonomic, the way that I remember that, autonomic is automatic. It's not voluntary. So your autonomic branch is sort of in a lot of ways, it's kind of the behind the scenes branch of your peripheral nervous system. So it is sensing information from, organs, sub-aware, not deliberate bodily functions. And it's also sending commands out to those things. Your brain has to tell your intestines to engage in peristalsis. Your brain recognizes when, for example, when your bladder is getting full, when you're hungry. None of these processes are deliberate or conscious even. So it's automatic. Somatic branch, I'm moving my muscles, I'm doing this on purpose. Totally different. Autonomic branch, tremendously important in the regulation of the organism, as you can imagine, because then you have the sympathetic division and the parasympathetic division of this automatic, kind of your basic operational functions. You don't have any sort of cognitive control over your sympathetic response, your parasympathetic response, and of course parasympathetic, your rest and digest function. You'll read in Stahl, you'll hear us talk a lot about sympathetic tone and sympathetic activation. And we're not talking about sympathy, but we are talking about really, really, really emotional. So I guess that's a way to look at it. But sympathetic is the branch of the autonomic, the division of the autonomic side of things. So the behind the scenes, automatic, not conscious awareness that comes to fore when you perceive a threat, largely. And that results in this whole activation of this whole system in your body to, what are some of the things that happen when the body perceives a threat? sympathetic activity increases. What might that look like in somebody? Because you see fight or flight here. Right, Karen, exactly. Your heart rate goes up. You might start sweating. Your breathing may increase. Your pupils will dilate to let in more light, to enhance your visual acuity. Blood gets shunted away from your guts, and it gets shunted to where you probably need it, which is going to be your muscles, to get you out of there, or to fight till you can't fight no more. Yes, so sympathetic activation is the behind-the-scenes response that is triggered probably not by conscious awareness a lot of times. And humans, mammals do this, reptiles do this. This is not unique to us as a species. Oh, contrary. Parasympathetic division. So if sympathetic's not being activated, then your body's really trying to be in homeostasis as much as it can. So the balance to that is parasympathetic division. When that is activated, your body's going to try to rest and recover from the last time it was sympathetically activated. And there's a lot of restorative processes that go on here. Heart rate goes down, breathing slows, blood makes its way to your intestines to try to digest whatever food you have, maximize nutrition, all those things. As you notice, the only neurotransmitters involved in the parasympathetic response is acetylcholine. In our central nervous system, and where your spine connects to little nerves exit the spinal cord, and it meets with a ganglia. So you have your spine, little nerves exit to go to their different jobs. Whatever nerve, maybe it's the nerve to go to your gut, maybe it's the nerve to go to your diaphragm, whatever it is. And acetylcholine is what gets released to say, okay, nerve going to my intestines, go, be active, go do what you do. And then once that nerve makes it to its destination, to the actual maybe, smooth muscle tissue in the intestines, it also releases acetylcholine there. So your parasympathetic rest and digest functions are really dependent on acetylcholine. You can see here how sympathetic is different because it ends up with norepinephrine causing the activity seen in sympathetic activation. But really important to file away that acetylcholine is the end-all be-all with parasympathetic activity, your rest and digest. So if someone's on drugs with really strong anticholinergic properties, you might expect an interruption or a decreased efficiency of parasympathetic activity. They won't see so well because they can't get their pupils to constrict back down when needed. Bowel will be slowed. Bladder will be slowed. Salivation will be slowed because this is autonomic. This is not automatic functioning. I mean, this is not deliberate functioning. And it's all acetylcholine, typically muscarinic acetylcholine receptors involved that gets blocked to have the can't see, can't pee, can't drool, can't stool, that little memory mnemonic right there. Acetylcholine is also critically important in cognition and in memory. So you would expect folks on anticholinergics to maybe have some trouble with memory, and also to maybe just feel a little foggy, just a little slower. We'll get to that later. The cerebrum, it's the outside, it's the bark, it's the cortex, it's the outer layer, not real thick. And anything that's gray is a cell body. Anything that's white is potentially a whole lot of myelin and insulation. So you got a gyrus, which is singular, of gyri or gyri, either way. You have a sulcus. Some people say sulcus. It doesn't matter how you want to say it. But when you look at the outside of the brain, it has a lot of grooves and folds. And when you see things like a prefrontal gyrus, they're talking about a bump. And when you see something like a central sulcus, that is a valley. Okay, so just terminology going forward. 4 main lobes of the cerebrum. 4 main lobes, divisions, like actually anatomical divisions of the brain. The frontal, okay, it's your forehead. It's the front of the front, probably from about the middle of your brain. If you put your hand, kind of measure halfway down the top of your head forward to your eyebrows, that's about your frontal lobe. Really important. That's where we exist for the most part. This is a new evolutionary development that is gigantic in Homo sapiens and not so big in the brains of lower ordered, less complex, less cognitively efficient critters on the planet. What's important about the frontal lobe is there's a lot of conscious awareness involved in this. A lot of your frontal cortex, thankfully, has connections with limbic structures and with sensory structures. So we have the capacity to control emotional responses and to control motor responses, largely because of connections the frontal lobe has going. They call it top-down processing, from higher frontal structures down. The prefrontal, which just means in front of the front, I mean, that's the very, very, very front, kind of eyebrow and back. That is the seat of the highest development that anatomically humans have achieved. That's executive functioning, reasoning, planning, delayed. Not necessarily a good thing when you think about it. Anybody heard of EPS? Yeah, nailed it. Exactly. Extrapyramidal side effects is what EPS is. So you have this voluntary motor area, and those of you who've worked with patients who've been probably exposed to antipsychotics acutely or chronically, you have extrapyramidal side effects. This is not deliberate conscious movement, like I want to pick up my cup, so I'm going to reach over here and I'm going to pick up my cup. Extrapyramidal system is designed to support deliberate movement, but it's not involved in any way in deliberate movement. The extrapyramidal system, its job is to maintain stability, to maintain postural tone at rest. You're sitting there, y'all probably sitting there at your little desks, and you have your computer, and you're not consciously aware of how much motor tone, how much muscle tone is required to just keep you sitting in that chair and to keep you still. But there is a whole system, unbeknownst to you, keeping you still and upright and not hunched forward or falling out the chair or shaking. That's your extrapyramidal system, which is ruled almost exclusively by, we believe at this point, that the extrapyramidal system, so postural tone, just sitting still at rest, we think this is a dopamine acetylcholine balance. That acetylcholine tends to turn off dopamine, Dopamine turns off acetylcholine. Chorea, Kelly is asking if chorea, like Huntington's chorea, is that an issue of an extrapyramidal dysfunction? It is, but in a different way than, because when you think of a movement disorder associated with psychiatry, what's a big one that you think of other than Huntington's? We mentioned EPS. What's another psychiatric? Yeah, exactly. Perfect. Thank you. Parkinson's. Yes, and Parkinson's is, what do you see in Parkinson's? It's people who are stumped over. They're stooped. Hopefully not stumped, but they are stooped. They have this forward lean when they walk. They have itty bitty little steps. They're very unsteady, and they tend to have a tremor at rest. Not when they're doing something, but at rest. So where dopamine is too low, what seems to be the issue is that with tardive dyskinesia and with Huntington's disease, there is an overcorrection to where the body becomes extra sensitive to dopamine, and then you have excess movement. So they are related, but Parkinson's is more due to a deficiency in dopamine, whereas tardive dyskinesia or Huntington's is hypothesized to be due to an overcorrection and therefore a hypersensitivity to what dopamine is available. So you have the opposite result in this excess movement. So extrapyramidal side effects. These don't affect people when they're trying to deliberately do something. It's when they're trying to remain at rest. So it's really a disorder of inhibition of unwanted activity. Because that's what dopamine does. In the striatum, right here, striatum. These are not frontal cortex kind of structures, but these, your basal ganglia, your striatum, which is composed of the caudate nucleus, and the putamen. The striatum is so critically important in impulse control, in reward-based learning, essentially in the selection of a motor program to do something you have been rewarded for versus not selecting this unrelated motor program. And by motor program, I just mean, your brain is stored with, if I want to tie my shoes, I lean forward and I tie my shoes and I grab the laces and I loop them around, blah, blah, blah. When I think tie shoes, I know, it's like, oh, let me grab that file. This is the instructions to doing that. I don't need to stand up. I don't need to yell and scream. I don't need to sit down on the floor. Those would be motor programs or sets of instructions that are not related to my stated goal of tying my shoes. Dopamine in the striatum is pivotal to filtering out what you don't need to accomplish the goal and selecting with enough, what's the word I'm looking for? Force isn't the right word, but you need sufficient neuronal activity, kind of sufficient tone to get the job done. You got to block out what you don't need, but you have to sufficiently activate the sequence of events to get done what it is that you want to do, which is Parkinson's. You have movement you don't want and an inability to have sufficient tone of activities that you are shooting for. Nigrostriatal tract, if you remember, or if you've heard at some point in the treatment of schizophrenia, the nigrostriatal tract is implicated, and we will get there later. So this is a support system. Some critical terms in here. Striatum. Another lobe, because we kind of talked about the frontal lobe, that deliberate conscious action lobe. The next is the temporal lobe. more so, a little more rear, and it's conveniently located by your ears. Anything come to mind when you see temporal lobe? Anything in particular? Temporal, you think time. Temporal, time, history, memory. Well, that's appropriate because the temporal lobe has auditory association areas, sure, but also probably its main role for our purposes is going to be that the temporal lobe laterally is where your memory, hippocampus and amygdala, kind of lower temporal lobe, and medial will be where the amygdala is located, and a little lateral to that's where your hippocampus is. You see hippocampus, you want to think memory, and to a large extent you want to think emotion. What's neat is that memories can be stored different ways. You may have the emotional component will get stored thanks to the amygdala's innervation with surrounding structures. But there's also a play-by-play of memories too when you think about it. You might remember, oh gosh, I remember how I felt. And that's totally separate from, it's almost like that memory can hit you quick and immediately, but then when you actually stop and ponder it, you might remember, oh, well, I remember how the weather was, and I remember where I was standing, and I remember who was there, and I remember what we did, sort of that, the script from the event. That's stored through the hippocampus, whereas that emotional, how you felt part is stored through a separate structure. The hippocampus is also necessary, temporal lobe generally is necessary not only for making memories, but for retrieving memories. So you go to a friend's house, you haven't been there in 5 years. As you're walking up, you sort of start remembering, oh, I remember that, and I remember that's where they parked the car, and that's where they had their, they used to have a fire pit out there, or I remember that color. Your hippocampus is necessary for you to re-access those memories to prove, as you're, sensorily you're seeing these things, it's coming in, it's being processed, but it's also being tagged as, oh, you've been here before. What's important with that is when people have experienced dissociation, either depersonalization or derealization, there is a breakdown in that exact process, that the temporal lobe, your hippocampus, isn't being activated, it's being rerouted, and it's being kind of cut out of the experience. So you have people who say, I know I've been here before, but this just feels weird. This just feels unfamiliar, or I don't feel real. I don't feel something is weird. So either the person themselves or the environment, something about it is unfamiliar because they're unable to retrieve that memory effectively. With Alzheimer's disease, very often the hippocampus has been just downregulated and downregulated and downregulated till it's no longer able to connect new sensory experience to lay down new memories, and it's not able to retrieve old memories so as to determine, oh, this is familiar, I've been here before, I've done this before. And that is what we see in Alzheimer's disease. People lose the ability to they call it rapid forgetting. The memory is just gone. It doesn't get recorded. And gradually over time, things that used to be very familiar become less and less and less familiar. The hippocampus is very sensitive to hormones. It has a lot of receptors to hormones and to neurotransmitters because your body has some systems in place to say, hey, this is really important. You need to remember this. that in the context of an acute stressor, stress hormones will be released, and it amplifies the acuity of the memories that are stored. Memories are stored much more clearly. You think about people who've experienced trauma, their traumatic memory may be stored with such precision that even a certain color of shirt on a person on the other side of the room, if that retrieves a memory involved with the trauma, that's able to bring the whole memory back of every single detail, where I was, what I smelled, what I felt, blah, blah, blah. So, trauma, we're designed to remember things that were dangerous and risky to us. That sympathetic activation, that fight or flight, norepinephrine has a way to tell your hippocampus, hey, this is important, you need to remember this, so you can avoid it down the road. So it has a survival mechanism, of course, but it also, you can overwhelm that system as well if chronically exposed to stress. And we're seeing that inflammation apparently is going to end up being proven to cause every disease, including Alzheimer's disease. And Alzheimer's is not just the issue of your hippocampus is T9. There's just general loss of acetylcholine neurons in the brain, which is so involved in cognition and memory. Parietal lobe, that's going to be the part of your brain on the tip, the top behind the tiara. This is primarily an incoming temperature, sight, hearing, all gets routed into the parietal lobe. This is how you sense what's going on. And in the very back, the occipital lobe, visual, receptive, and association areas. From a psychiatric standpoint, the areas that we will focus more on, especially at this stage of the game, is going to be frontal processes and temporal processes and functions, because that is just more relevant really to what we do. That, and then, because this is all still tip-top Homo sapiens, highest order of development of any brains on Earth. You may have also heard of our mammalian brain and our reptilian brain. So we're just talking about the human brain thus far. Most, if not, I'll just say most of the action, and this often gets neglected, is activities of the mammalian brain. And that's when we get into, you see down here talking about, this is cocktail party stuff. Right brain people are more ethereal and non-concrete, and they're musical and creative, and left brain people are more into math and verbal. That's totally fine. There are a lot of studies that will actually rule out people who are left-handed, which I think is very interesting. But we're getting into the mammalian concepts, which the limbic system, this is above, it's higher than your reptile brain, which is common to mammals, and some of them are also present in reptiles and such. But when you see limbic system and you think, you can think emotion, you want to think learning, you want to think response, selection. None of the limbic system here this is not conscious, this is not deliberate. This is behind the scenes machinations of our organism that greatly influences our conscious awareness that we're really not aware of most of the time. Limbic system, we're going to talk about that a good bit. This is not a location, it's structures that tend to work together for a purpose. And when you think limbic, you're probably going to want to think generally the experience of emotion, the regulation of emotion. The cingulate gyrus is sort of a bridge between your frontal cortex and this limbic system that's involved in the processing of emotion and how to respond to it, what my response options are. The frontal cortex, I mean, that's a high structure. That's as advanced as we get, right? What is its involvement in the limbic system? The frontal cortex has a kind of check and balance role with emotion, that structures in the frontal cortex, specifically the dorsolateral prefrontal cortex, its activation increases on brain scans when people are starting to get, very upset or very angry or very scared. Not that the frontal cortex is contributing to that, but it speaks to these attempts to maybe kind of regulate it, sensing there is some activation happening that needs to be, whoa, we need to consider consequences. And now, wait a minute, what happened the last time I did that? Or what are going to be the implications of if I do this? So the limbic system, it's not one spot. It is just a group of structures that tend to activate together in the processing of emotionally charged material. And so it involves, your hippocampus trying to retrieve a memory similar to the last time that this occurred. Do I need to launch a fight or flight response or do I not? Hypothalamus, let me see, is the hormonal environment compatible with the current situation and the demands of it? Do I need to release a little cortisol? Do I need to pull back some cortisol? Are things wonderful? So I'll release some oxytocin. Limbic comes up when we talk about the mesolimbic tract in the context of schizophrenia. Because nigrostriatals, we're going to discuss that a good bit, the mesolimbic tract and its role in positive symptoms and mood lability, and really difficulty with response selection. The thalamic nuclei, just a bunch of neurons linking up together in the actual brain. Hypothalamus, this is that's basic right there. That's food, water, maintaining temperature. Am I hungry? Am I thirsty? Am I hypertonic? Do I need to drink some water? Am I diluted? Do I need to release water? Basic stuff. But as you'll notice, dopamine and serotonin are two primary neurotransmitters involved with the communication. Hypothalamus. The thalamus is where all incoming, Grand Central Station, I call it, all incoming information from baroreceptors to mechanoreceptors to thermoreceptors, it all comes into the thalamus, which is good because that allows the hypothalamus to have ready access and to respond quickly when there is some irregularity in homeostasis, to maintain the right temperature, the right hormone levels. Two in particular that we care about is prolactin inhibiting factor. So the molecule, neurohormone that gets released when lactation does not need to occur is dopamine. Dopamine is also known as prolactin inhibiting factor. Antidiuretic hormone, which is helpful when you're kind of dry and you don't need to lose any more fluid. Many SSRIs will oppose antidiuretic hormone. So when you see the syndrome of inappropriate antidiuretic hormone secretion as a risk factor with SSRIs, SNRIs, that's because those medications can limit the effectiveness of your pituitary controls. So that's why you check sodiums whenever people are on, really you should on any serotonin-related antidepressant. But we'll get down there later. Here's a picture. Oh, reverse. Here's a picture of the brain. There's that central sulcus. There's the tiara. Here's your parietal lobe back here, occipital way in the back. Frontal lobe, kind of from your eyeballs would be here if you could see them. Frontal lobe, grooves, bumps, sulcus, gyrus. And you see how far in the front, where your orbital frontal cortex would be about there, dorsolateral prefrontals would be on the side, but imagine roughly here, but going away from us. Versus how much closer, they call it limbic lobe, that doesn't actually exist. But, I mean, look how much closer situated things like, okay, so here's your little amygdala and hippocampus over there, and there's your striatum is roughly in this area. It's not listed here. But it speaks to the much quicker connections between these older structures, these mammal kind of reactive, responsive structures, whereas you're reasoning, wait a minute, impulse control, consequences, look how much farther that is away. When you think about how quickly these impulses travel with all of that myelination, we're really designed to be more automatic than pensive and deliberate, for sure. The cerebellum, which is this guy right there, it looks like a little miniature brain. It does a lot of processing of balance and fine-tuning. When you think about I think it's proprioception, that is your awareness of your body in space. That's another subconscious, tremendously involved processing system. So when people have issues with balance, you think about folks who look intoxicated. Their movements are not coordinated. Their legs are flopping around. They're kind of leaning, and it's just sloppy looking movement. Balance is off. People with cerebellar lesions or a cerebellar stroke will have a lot of issues with balance and more problems when they're trying to be deliberate than not. Extrapyramidal symptoms are different. It doesn't look sloppy. It's actually very restricted. It's too little movement. It's a tremor when the person is trying to rest, actually. But sometimes the extrapyramidal symptoms will go away if the person is actually, like say they have a tremor to their hand, but if they reach for a pen or reach for a cup, the tremor is resolved. Because a competing motor program is more powerful than the one that remains, than the one that is not being filtered out. So if you try to actually do something, well, that trumps the unnecessary movement when there's nothing else competing with it. As opposed to, like when you see someone who's had a cerebellar stroke, it doesn't matter if they are trying to do something. Okay, y'all, we're almost getting to the end. Can y'all hear me okay still? Okay, great. Thank you. Thank you very much. My computer was telling me it couldn't. So lower frontal, the cortex, high order, limbic system, mammalian, kind of in the middle. And then we get to the brain stem, which is the lowest, oldest part of our brain. This is responsible for basic survival functions like breathing, like maintaining a bit of blood pressure, like throwing up when exposed to a toxin. These are things that even a person who is in a coma or a person who is being anesthetized, these functions are still active. So this is the most basic, it's the last ones to go, on the lowest, oldest part of the brain. The midbrain is important for our purposes because that's where the origination of our big neurotransmitter players come from. The substantia nigra originates in the midbrain. The locus ceruleus originates in the pons. Pons just means bridge. It's the bridge between the midbrain and the medulla, which is kind of the lowest part of the brainstem that eventually transitions into your actual spinal cord. Really old, really basic. The medulla is also where the lateralization, the right part, the sensory processing of the right part of your brain and the right motor actually controls the left side of your body. It's the decussation of the pyramids is where it crosses over in the medulla. Centers for respiration, like we talked about, blood pressure, vomiting. Neurotransmitters involved in communicating nausea and vomiting are largely serotonin and dopamine. It's why Zofran helps with nausea. It's why Reglan helps with nausea. Another reason why people might get nauseated when they're taking things that boost serotonin. And the Rafe nucleus, which is the origination of serotonin, the main serotonin neurons, where they then branch forward and go to the rest and have all of their connections. So the midbrain, it speaks to how integral serotonin and dopamine and norepinephrine are, just a basic foundational physiologic functions. It's not just depression, it's not just anxiety, it's temperature and hunger and nausea and reinforcement learning and pleasure and reward. So we want to be alert to the many other functions these neurotransmitters have in addition to helping people be less anxious. Cranial nerves are important, in particular these four, because of their relevance to side effects and the medications that we prescribe. Now, cranial nerves, they originate from primarily from the brain stem. They're not in the actual brain, they're not in the actual spinal cord, so they're peripheral nervous system structures, which has those two divisions, one of which is parasympathetic, which we mentioned just a little bit ago, is so dependent upon acetylcholine for proper transmission and functioning. So when you block acetylcholine, particularly muscarinic receptors here and there and everywhere, you're blocking these cranial nerves attempt to conduct, their basic operations. Like you need your parotid glands, and you need this facial nerve to tell your parotid glands to release some saliva, right? So if you block that process, then you'll have a really dry mouth. If you block muscarinic receptors, specifically in respiratory secretions, will go down. You'll just really, the dry mouth is twice attacked by anticholinergic drugs. You will have an inability to constrict the pupil, which, if anyone has ever taken Benadryl and had that happen to them, you will think, oh my gosh, I cannot see. You really can't see to save your life. Fascinating. Happened to me one time. I hope it never happens again. But I thought, oh my gosh, this is what they mean. I can't see. And it was dark, so I really needed to be able to see. And last but not least, the vagus nerve. Cranial nerve 10. It's not cranial nerve X, it's the vagus nerve, and it's cranial nerve 10. This guy, wow, is in charge of a lot of functions in the body. Not only just to not only in the regulation like we have here, blood pressure and heart rate, but this innervates cardiac system, GI system. This is intimately involved in maintaining the hormonal environment, stress hormones, the health of the gut. If you've ever vagaled down, or you've ever worked with patients who've done that, it's just this really quick drop in heart rate and blood pressure. The vagus nerve actually innervates the your AV node, your atrioventricular node, which is sort of the midpoint between the atrium chakra and the ventricle, specifically on the right side, where your SA node is trying to cruise on through and initiate a heartbeat. Your vagus nerve innervates the AV node and is able to directly right there slow down heart rate. But if you're blocking the vagus nerve's attempt to slow down heart rate, well, you put the patient at risk for increased heart rate. Something you try to do in a code, at least that we used to try to do, is give atropine, which is a really, really strong, I'll say it's a really strong, it's just a potent anticholinergic, because you're trying to block your vagus nerve's attempt to slow down anything. Let every cardiac impulse go through. So when you're writing, you have somebody with an extensive cardiac history, and maybe they have a longer QT interval as it is, you want to be really careful writing drugs that might speed up heart rate and even further extend QT. So just keep these cranial nerves in mind. This is acetylcholine issue, parasympathetic issue. Kind of hit this already. You're born with your brain being about a fourth its eventual weight, and your brain is as heavy as it's going to get by the time you're 11. So that's a lot of development that needs to happen. 1 trillion support cells in the brain at birth. That is nuts. Really pretty wild. Almost done. This is just a description. This is a neuron. This is the soma. Okay, this is the body. This is where the nucleus is. This is where all of the metabolic activity of the neuron occurs. These are the branches. Each of these connect to other neurons, receive input from other neurons. So when you read install the somatodendritic end of the cell, they mean this side. It's where the soma and the dendrites are. It's the incoming side. And then you got the axon, this big long part where the electrochemical energy, the action potential travels down. We're going to talk about the electricity later. We're not going to do that tonight. But it's all sodium-potassium pumps. It's all sodium and potassium swapping along this whole axon that makes the electric impulse travel down to the end where the terminal is, the axon terminal. So this is where, once the impulse makes it all the way down, the electricity makes it down to the terminal end, it results in a physical change in the cell walls here at the terminal, and neurotransmitters get released. Vesicles merge with the cell wall. It releases into the synapse. The synapse is here. It's an actual space between where this nerve terminal ends and the next neuron begins. This happens lightning fast. So somatodendritic end is here. This is very relevant when you're reading about the 5-HT1A receptors on serotonin neurons are here. They're somatodendritic. But then you have presynaptic receptors. And a receptor is just, all it does is receive. That's all a receptor does. It receives a neurotransmitter from somebody else. So you also have receptors on this end. I hope you can see my little pointer. Presynaptic receptors. So neurons may be receiving neurotransmitters like right here at the end. When you read about serotonin, for example, 1B/D receptors, those are autoreceptors on the presynaptic end. So it's still on this nerve. And then you have postsynaptic receptors, which is the receptor on the other end of the synapse. And this could be any neuron over here. Anyone. And the way that these cells communicate is by sending chemicals from me to you, from this one to that one. And those neurotransmitters initiate some sort of chemical change or functional change in the next neuron, and it passes the message on down. The main ones we're going to talk about: acetylcholine, you have your monoamines, GABA-glutamate, peptides. Peptides, yes. We're not going to talk about them because they are not relevant to our purposes at this point. We have plenty to talk about with these 6 here. So we say that to say there are other neurotransmitters other than the main 6 we talk about. Currently, they're just not really implicated in the treatment of disease. So what neurotransmitters do? They land on a receptor. If a neurotransmitter, if it functions as an agonist, then the receptor is activated. And whatever that receptor's function is, it will do more of it. A neurotransmitter could be an antagonist, or a drug could be an antagonist of a receptor. It may reduce whatever activity that receptor is responsible for. You also have partial agonists, which means they kind of do it, they kind of don't. Autoreceptors. All of this information is in stall. What an autoreceptor does is once a neurotransmitter lands on it, all systems stop. It's kind of like you've reached capacity, we're not going to do anymore. Real important. Sodium Sodium is a cation. It's a positive charge. So if sodium enters a cell, it depolarizes. Chloride is an anion. It's a negative charge. If it enters a cell, the cell is less likely to fire. Basic chemistry stuff. GABA enhances the influx of chloride. So GABA is going to be calming to cells, makes them less likely to do anything. You think GABA, you think anxiety reduction. No surprise, it lessens the activity of a cell. So it might turn on a receptor, turn off a receptor, meaning increase or decrease the activity of the neuron downstream. Last slide, the end. Really important slide. Kind of a summary. We talked about the 6 main neurotransmitters implicated in psychiatric disease and treatment, where they come from. And at the bottom, because a lot of people want to know, I want to get better with the drugs, I want to get better with the drugs. Why pick this one versus that one? We start that now. The way that drugs help is through primarily these mechanisms. If a neurotransmitter is not doing what it's supposed to do, and that's resulted in some kind of detrimental functional change in the brain, causing depression, causing panic, we can add really artificial neurotransmitters, a la drugs, psychiatric medications, to try to correct what's broken. You can block a receptor, you can block the reuptake of a neurotransmitter, like Prozac, that's what it does. You can slow down metabolism, that's how MAOIs work, that's how Aricept works. You can stimulate a receptor, you can make a cell release more of its neurotransmitters, like a Adderall, that's what it does. You can manipulate autoreceptors, turning them on, turning them off. So going forward, drugs are trying to correct whatever is not in balance, whatever's not functioning properly. That's what we do. And we focus on these 6 neurotransmitters and the respective receptors for them. And thankfully, for all of our sakes, that's it. That is it for tonight. Sorry we kept y'all. We got a 4-minute late start and we went 10 minutes late past that. We're going to continue next week with pharmacokinetics and pharmacodynamics, so more of the study of the drugs in the body. Morgan Todd is asking a question about intake note grades. I encourage you, Morgan, and anyone else who's listening, to check in with your clinical instructors and ask them when they anticipate being able to get you that feedback. That's a really good question. Direct it, send it on over to your instructor, most definitely. So there's a ton of information. Please listen back through to the lecture and read the readings and take notes on this PowerPoint. Any questions? You're welcome to ask them now. You're welcome to email me later. But this will be uploaded shortly. We appreciate y'all's attention. This stuff is pretty dense, but it's important. We're going to keep revisiting it.
Simone, so next week we're going to talk more about the neurotransmitters. Is this a good slide to start on for next week?
There's no reason why we can't.
Okay. It just seems like you covered a lot of information, and it just would be nice if you repeated what you talked about in this slide as a way to move on.
We can segue. We can segue right on into next week with this slide. Absolutely. Absolutely. All right, everybody. We'll go rest and digest, and email us questions. We'll see you next week.